Chapter 8, Section 2, Nucleic Acid Structure. Nucleic acid. When we look at the structure of nucleic acids, turns out there is some complexity above just, hey, look, it's a strand of nucleotides. Just like we talked about with proteins, here we're going to see different levels associated with things: primary, secondary, tertiary. Primary structure, basically it's just going to be the nucleotide sequence, AGCT, AGCU, DNA, RNA. Secondary structures, well, sometimes there's going to be structures due to what nucleotides are there. There are going to be some twists, some tweaks. And if they start to fold, do more folding in on themselves, well, you're going to get this tertiary structure, which we're going to see formed in tRNA, or ribosomal RNA, where instead of one long strand, it's going to form little loops and curls that allows for complementary base pairing of RNA nucleotides with different portions of the same sequence. We have to keep in mind that a lot of what we know about the DNA double helix, yeah, Watson and Crick, you've learned that since you were kids. There's also been this push to say, hey, we also need to shed some light on, some of the information that Watson and Crick used. Unfortunately, there's been a big push to vilify Watson and Crick. Yeah, look, they were a-hole people, all right? Yeah. Quite intelligent. They did quite a bit of work on their own. It's just that the one link in the chain they were missing was this X-ray crystallography right here. This is an X-ray crystallography that was done in the lab of Maurice and Franklin here. Rosalind Franklin was her background, she was a specialist when it came to X-ray crystallography. Remember X-ray crystallography? You take some organic or inorganic compound, you make it a salt, precipitate it out as a salt, forming a crystal. You take that crystal, you bombard it with an X-ray beam, and then you look at how that disperses as it hits an X-ray film, X-ray crystallography. And then based on the dispersion pattern, you back calculate the structure of your salt crystal, okay, your crystallized organic or inorganic compound. Franklin was able to do this. She got this, a semicircular pattern with an X in the center. She couldn't figure out what it was, okay? Some people are now trying to, do a little revisionist history and say, oh, she could have figured it out given enough time. But the thing is, it wasn't just, these two people here, Franklin and Maurice, who are working on this. It wasn't just Watson and Crick. There were many, many other labs around the world who were trying to figure this out. Remember Chargaff and Chargaff's rule: for every A, there's a T. For every G, there's a C in the double helix. He couldn't figure out how that actually worked. Based on his work, Chargaff and some other labs had said, oh, it's just a single strand that goes, if you see an A somewhere there, you're going to also have to see a T. If you see a G somewhere along the strand, you're going to have to see a C. And they just kept coming up with these ways that just didn't fit. Well, you have this X-ray film. Now, exactly how it came into the possession of Watson and Crick, well, they say one thing, her supporters say other ways, but it did. It got into their possession. And then Watson and Crick had figured out, okay, we know the DNA strands. It's two strands bound together somehow. Okay. Other labs were sitting there saying, yeah, it's two strands next to each other, A to A, T to T, G to G, C to C. The problem is that made this structure that didn't fit this X-ray film of a nice circle with crisscross. Well, yeah, Watson and Criss figured out that based on Chargaff's rule, for every A there's a T, for every G there's a C, and that you have to fit within this constraint right here. You have a double helix. Two strands of DNA, but instead of being parallel, they were anti-parallel. Boom, 5 prime, 3 prime. 5 prime down here, 3 prime up at the top. If you look at this end on, the ribbons you see here are the sugar phosphate backbone. To picture this in your head, think of a ladder, okay? The rungs, and then you got the risers. If you were to take that ladder and not twist it, but wrap it around a flagpole, and then look at it end on, what you would have is a circle where the risers had wrapped around the flagpole. And when you looked at it end on, the rungs, the nitrogenous bases, are going to form this X pattern in the center. They figured it out. And the thing that they were able to do that no other labs were able to figure out at the time, and to us, we sit there and go, well, duh, A to T, G to C. 3 hydrogen bonds holding the GC together, 2 hydrogen bonds holding the AT together. You've seen the pictures in textbooks where Watson and Crick are looking at their little wire, ball and stick molecule that they, model they made of this. Yeah, if you took it and put it end on, you saw the circle, and nitrogenous, the A to T, the G to C, made the X. Well, we also have now taken it beyond that. And we have noticed that the rungs are evenly spaced. They're evenly spaced 3.4 angstroms between them. Okay, angstrom, this is going to be a really stupidly small relative distance thing. The circle that, Franklin's X-ray crystallography showed that, Watson and Crick's model when it went boom. The circle has a diameter of 20 angstroms. And this whole idea, instead of taking a ladder and just twisting it, but wrap it around and then look at it end on, meant that you would end up with different sets of grooves. A major groove and a minor groove. Major groove, minor groove. And if you start at one point, you go through the major groove and through the minor groove so that you do a complete 360 turn down that sugar phosphate backbone, boom. That distance is 36 angstroms, which is roughly 10 rungs. 10 rungs, 3.4 between, have to add in the physical distance width of the nucleotides themselves, the nitrogenous rings, and the rungs, comes out to be just under, round up 36 angstroms. 10 times that, 34, add in the distance of, there you go. Here you see a computerized ball and stick model of what Watson and Crick put together and what they figured out. Nowadays we do this space filling model. The purple, I'm sorry, not the purple, the blue with the little reds inserted here and there, that's the sugar phosphate backbone. The yellow, those are the nitrogenous rungs. A to T, G to C. 3.4 angstroms between, and to go from here to here, one complete 360 turn, 36 angstroms. From this side to that side, the complete diameter, 20 angstroms. If they were parallel, which some labs are, at the same time as, between Chargaff and Watson and Crick. So we're talking mid 40s to Watson and Crick in the late 50s, I think it was. They were trying to say it's parallel, and it just never worked. Because if they're parallel, the nitrogenous rings for both rungs, are both pointed the same direction. So if you flipped them and then tried to do it, now they're oriented in the wrong way, and instead of being nice or flat, they're, more disjointed. Couldn't work. Watson and Crick figured it out. Instead of parallel, anti-parallel. It allowed the nitrogenous rings to be almost flat compared to the double helix. Also put them in such a way that they were oriented towards each other at just the right angle. Like I said, may have been shit people in private, but crazy smart. One complete turn, which is about 10-ish rungs, 36 angstroms. What's very important also is by going by a Watson and Crick's model, as we see here, that we meet Chargaff's rule. C and A, there's a T, C is C, there's a G, 3 hydrogen bonds, 2 hydrogen bonds, but they're complementary. Okay, we call this complementarity. So technically, I could sit there and tell you, here's the DNA strand, 5' CAA TCG TCA 3'. Just reading that, you can deduce what the other side's going to be. 3' GTTACG blah blah blah. Wherever you see a C, the other strand has a G. Wherever you see an A, the other strand has a T. It just works. The textbook says the hydrogen bonding does not contribute significantly to the stability of the structure, which is quite at odds with what I was always taught in genetics, what I always teach in genetics and cell biology and intracell. This aids in stability, holding together the double helix. Yes, it's twisted around. You got the hydrogen bonds, the twist, holding it together, and in your cells you have the DNA double helix wrapped around histones and all this stuff, which is why we can get it from 50,000-year-old frozen mammoths. So I don't agree with that. The biochemists are going to sit there and say, and they're right, that it's the negative charges of the backbone. And there's going to be cations that will stabilize the backbone, so the backbone doesn't twist, doesn't bend so much that it shears, or that it's going to do like we saw with the RNA oligonucleotide, where it can, spontaneously form cyclic, and parts of it just pop off. The reason why I don't always agree with them on this is this bullet right here. From a biologist standpoint, from somebody who's done a lot of genetics work, we know that, they're sitting there saying, okay, it's the sugar phosphate backbone. The sugar phosphate backbone's going to be the same, because it's the same sugar, it's the same phosphate group, forming the same phosphate-ester bond. The difference is going to be in the middle of the double helix, where you have the nitrogenous bases. A sequence in an oligonucleotide or a polynucleotide that is what they call GC rich. Lots of G's, lots of C's, so you have the 3 hydrogen bonds, is going to hold together and be harder to pull apart than a sequence that's AT rich. Lots of A's, lots of T's. It's the same sugar phosphate backbone. Why would it be harder? Why would it be more stable if the hydrogen bonding had nothing to do with it? So from a biologist standpoint, I don't agree with this. This was written by biochemists. They're coming at it with a different take on the same data. Welcome to the interplay between chemistry and biologists. Who's right, who's wrong, depends on how you want to interpret the data. Depends on what you're using to measure. You know, what's your frame of reference? As you've learned in Intro Cell and Molecular Cell Biology, genetics, you know, Watson and Crick also figured out this whole complementary base parent, or complementary, going back, not complementary, semi-conservative DNA replication. Once Watson and Crick figured out here's the double helix, people came along, okay, we're going to take this information and run with it. They're trying to figure out how the DNA double helix can be replicated. New strands made, new chromosomes made. There was lots of different ideas, lots of different theories. Watson and Crick, rightfully so, figured out what they referred to as semi-conservative. The original double helix will be pulled apart by enzymes. Enzymes will pull it apart just enough so that DNA polymerases, specifically DNA-dependent DNA polymerase, can come in and start reading the blue, which is the original, and making the new, the red ribbon. Why is it semi-conservative? Well, because the blue, which was the original double helix, is now part of the two new double helixes. Makes sense. This one has no errors, no issues. Then the two new ones, which are based on this, will also not have any errors, will not have any issues. But, as we've seen in all the other chapters, there's the textbook perfect, and then there's real world. Turns out the deoxyribose in DNA has some different configurations, because remember, as we talked about in the last chapter, with the sugars, all the carbons in the sugar can act as chiral centers. Hydroxyl pointed up, hydroxyl pointed down, that type of stuff. You know, as they're twisted around the chiral center, it means that the sugar itself is going to bend, twist, and tweak. And whether it's the 3' carbon going up or the 2', 3' going up, 3' endo, 2' going up, they call it 2' endo. Different shape to the sugars, which means the sugar phosphate backbone is going to have some slight variation in how it's set. Also, I have the nitrogenous rings themselves. Okay, they're not static. They swivel around based on that carbon-nitrogen bond we see right here, that N-beta glycosyl bond we talked about earlier. So it, just flaps around back and forth, especially when it's in its free nucleotide state, or as the nucleoside. Once it gets put into a double helix, it's set. It's not going to be able to swivel. It's not going to be able to move much because it's complementary base paired, A to T, holding it in that place, in that orientation. Turns out that the double helix that Watson and Crick figured out is not the only way it is. I mean, though, what they now refer to as the B form of, this is the most stable there. in physiological, where everything's, in the cell, in the nucleus, in a cytoplasm, if you're talking prokaryote, this is what it's going to look like. Okay? 3.4 between the rungs, 36 angstroms, 10.5 rungs per turn, 20 angstroms wide. Perfect. Until we started looking at more and more double helices, and we developed better and better mechanisms beyond just the crude X-ray diffraction, X-ray crystallography that Franklin has used. Fast forward after Watson Crick, another decade or two, and they started coming up with better and better mechanisms, better means. And they started to figure out that there's more than just what they now called the B form. The A form, which is still right-handed, but instead of being 20 angstroms wide, it's just a little bit wider. Things are a little more relaxed. It's not quite as tight of that turn that we see with the beta, or I'm sorry, with the B form. It's a little looser and it's, spiral. Thus it's a little wider. Instead of 10.5 rungs per turn, it's 11. So it's not a whole lot wider, but it is just enough. Think of it as more relaxed right-handed twist. B form, aqueous solutions. A form, non-aqueous solutions. If you isolated the DNA and you say put it in alcohol, there you go. Put it in some other solution that's not water-based. A form. But, there's always a but, there's another form. A left-handed twist. The left-handed twist, which is completely ass backwards to the A form and the B form, we call this Z form. The A form and the B form, even though A is a little looser in its turn, its right-handed twist, than the B form, it's still a nice, smooth thing. Z? Yeah. Those major minor grooves that you see with the A and the B forms, they're still there, but yeah, not quite as smooth. So you look here, A form, B form. B form, Watson and Crick, okay, major minor groove, 20 angstroms across, blah, blah, blah. Here, yeah, a lot looser, a little bit wider. The nitrogenous rings, you notice also, because it's a little looser, a little wider, they're not quite as parallel as you see here. They're a little more tilted, a little more disjointed, but they're still there. Then you look at the Z form here. The Z form, it's more of a zigzag, which is why they call it Z. And instead of the right-handed twist, it's a left-handed twist. And everything's a little ass backwards. It's a little harder to get into this form. This form has its own biological functions. It's a little more harder or it's actually a lot harder for enzymes to access this. It's going to be a lot harder for interactions with other nucleotide, polyoligonucleotides, RNA strands, or anything like that. You notice here in the box, talking about this A form. It's a little looser in its turn, and it's squashed a little bit more. So it's a little loose in its turn, but it's not like it's stretched more. It's now been squashed down more. 11 as opposed to 10.5 per turn. 2.6 as opposed to 3.4. And you look at the Z form, completely the opposite. It's now even narrower than the B form. The base pairs, now 12 angstroms, so they're really stretched out. So this going to the left-hand turn really stretches things out, makes it narrower. And as you look here at the end on, not all the nitrogenous rings are going to be pointed in. A lot of them are going to be pointed out. And it's just going to make it more difficult for the base pairing to occur. It's going to make it more difficult for things to get in, to access, to be able to utilize whatever information may be there. Remember at the beginning of this section talked about there's going to be, levels of complexity, primary, second, tertiary. Well, some of the complexities we see there are going to be due not just to the shape, B, A, B, Z, but also what sequence the nucleotides are in. We see what is referred to as palindromes and mirror repeats. Palindromic sequence, TTAGCAC. It's the same CACGATT. It's a repeat of this, just backwards. So these palindromic sequences, spoiler alert, this is where you're going to find the beginning, that region right before a gene, where you want to have different transcription factors, associating with, recruiting in RNA polymerases and things like that. They're going to focus on these palindromes. You're also going to get these repeat sequences, where it does some of the same thing. Look at this, TTAGCAC, CACGATT. It's the same thing on the same strand. So when people ask what's the difference between a palindrome and a mere repeat, palindrome, look, they're on opposites. AATCGG, AATCGTG. Whereas here, it's on the same strand, the same DNA strand. The opposite side here that's not highlighted has the same thing going on, because remember, they're complementary. Single-strand nucleic acids are very unstable. We saw that, when we were talking about the RNA and how it will spontaneously hydrolyze, and the released nucleotides will become a nucleoside that has the, phosphate cyclically added at the 2' and 3' carbon. Well, to keep things like that from happening, double-stranded nucleic acid is just inherently more stable. So you take, here they're showing a single DNA strand. This could also be, if you replaced all the T's with U's, and this would be RNA. RNA will do the same thing. It will form these hairpins. I know, most people don't know what hairpins are anymore. But back in the 50s and 60s, when they were first starting to look at this stuff, women still used hairpins to hold their hair in place. A hairpin has this long shape. It would have this little, and then it would go like this. That's what a hairpin used to look like, for those of you who don't know. Well, when they first figured out this structure, it reminded them of this hairpin. This single strand struck out like that, whether it's DNA or RNA, is inherently unstable because, remember, it can twist around that phosphodiester bond. If it twists, it can bend. If it bends too much, things break. The nitrogenous bases are also, forming tauter, going through that tauterization, and they're, unstable, switching between these different forms. But here, as they are complementary base paired, no swiveling on the phosphodiester or the sugar phosphate backbone, no tautomerization of the nitrogenous base because it's hydrogen bonding with something that aids in stabilizing it. Whether it's DNA or RNA, this is inherently more stable than this. The double-strand DNA you see here, this nice straight, well, this is more stable than this. If it goes through and it bends in on itself and forms this cross shape, inherently more stable. It just is. So that's why at the beginning of this section I was talking about there's these levels of complexity where these things can build on themselves. These structures will, by forming this, if there's a gene that's just behind this, this will inherently interfere with any transcription factors recruiting in, or promoters recruiting in and transcription factors. The promoter transcription factor, bound at a palindrome right in here, would stop. Having this hairpin there would stop those, promoter transcription factors bringing in a polymerase and the making of a messenger RNA. Now, it was figured out in the 60s that the Watson and Crick B form, what we now refer to as the B form DNA, has the potential for alternate base pairing. Alternate in that, excuse me, instead of 2 strands anti-parallel, you might be able to insert a 3rd strand in there. Not a complete length of the chromosome, but maybe in short segments. Now it's a little unsure. I'm a little unsure of this because I've seen it talked about both ways. Here's what I'm talking about. Is that it's the true B form that has been slightly altered, still has the anti-parallel, but you offset the bases just enough, offset the strands enough, you can insert a 3rd strand in there. And as you see here, whether it's a TA or a GC, the hydrogen bonding will allow for the 3 nitrogenous rings to interact. I've seen it discussed that way, but I've also seen it discussed that it can't be the true B form. It has to be 2 truly parallel strands of DNA. So I'm not really sure which it is, because I've seen it discussed in primary literature both ways. So it raises the question, does this actually happen in situ? Or in vivo, in the cell? Or is this just an in situ, or I can't remember my Latin. Does it happen in the cell, or is it just something that can only occur in the test tube under certain conditions? Well, they've also taken it a step further, and they found that, hey, guess what? 2's great, 3 can happen. We can also make it happen with 4. 4 strands of, nucleic acid that are going to be, rich in guanine, GC residues. But it's going to be in such a way that instead of the GC directly, it's going to be G to G, as you see here. So these strands, these 4 strands, are going to have not have complementary base pairing, but G-rich regions that are the exact same sequence. They can be truly parallel or they can be anti-parallel, and this can still occur. Okay, this one I know for a fact is going to be test tube only. And when you look at our RNAs, the one we always think of first, messenger RNA. always use the analogy when people are kind of can't figure out what messenger RNA is truly used for. You go to the library. You need information from a textbook or from a book. That book is 10,000 pages long. It's big, it's thick, it's heavy. Well, you can check out and try to, haul around that 10,000 page book, or you can go find the nearest Xerox machine and make a copy of the 1 page of information you need. Leave the book, take the 1 page. That's what messenger RNA is. It's a copy of that one out of the 10,000 plus genes we have in our chromosomes. It's inherently unstable because you want to have a short shelf life on messenger RNA. You do not want this sticking around for long periods. You want it there long enough for a few ribosomes to start at one at the 5' cap and read down to the poly A tail. In enough polypeptides made that you're good to go. If you need more, you can just go back and redo transcription again. Up till now, whenever you've talked in intro cell and molecular genetics, maybe even cell biology, when we talk transcription, we always are a little vague on just how many genes are being transcribed. In us eukaryotes, 1 messenger RNA, or 1 gene, 1 messenger RNA. Okay, they go together. We refer to that as monocystronic. But in the prokaryotes, they do it a bit differently. For them, they do what is referred to as polycystronic. If 1 gene is required, or you have a set of genes that need to be required to be transcribed at the same time and then translated into polypeptides, because they all seem to work together towards one goal, say like the lac operon, the transport in and the metabolic utilization of lactose. Well, why sit there and have to go through transcription 3 or 4 times on 3 or 4 different genes and hoping the timing is right when you can just transcribe them all at once? 1 long messenger RNA that has the 3 or 4 genes on it, polycystronic. Translation is going to occur. Ribosome is going to hop on at the 5' end, and it's just going to march on down. Boom, boom, boom. Here's this, the new start codon. Here's the first polypeptide. Keep going down, down, down. Here's the next start codon. Here's the second polypeptide, and continue on like that. In textbooks, we always draw messenger RNAs and molecular RNAs as just these straight lines. And a lot of times students just incorrectly infer that, messenger RNA doesn't have any structure to it. It's just this straight line stuff that's going to fall apart all on its own. Couldn't be further from the truth. Even if it is just a single strand of nucleic acid, we find that the RNAs, messenger RNA specifically, is going to have a slight twist to it. So not all the nitrogenous bases are all pointed up in one direction. Okay, by having that twist, it gives a little bit more stability, as you see to the picture right here above my head. The the nitrogenous bases are going to stack, boom, boom, boom. So they're, pushing and pulling against each other, to keep everything kind of spaced out equally. And as we saw before, as I talked before in a previous slide, this long strand that's got this slight twist to it will bend back in on itself, form those hairpin loops, which allows it to have greater stability. And what was a 2-dimensional line will now have 3-dimensional structure. Has 3-dimensional structure, there could be a lot of different functions. So when you go and you look at different RNA strands, they can bend in back on themselves for those hairpins, or two different strands can come together as we're seeing here. Here's one strand, starts here, goes up, forms its own little hairpin. This is a nice little three-dimensional structure. An internal loop, bulge, and then boom. Here's another one, base pairing with it. Wherever you see a C, there's a G. Wherever there's an A, there's a U. Because remember, U, because it's RNA. This bulge here, without a base pair there, means it's going to have a bend in it. The bend, another loop, and off you go. This inherently stabilizes the RNA structure. Okay? Just like we saw with the polypeptides. Primary structures, the amino acid sequence, secondary structures, alpha helices and beta strands. Well, here's the nucleotide version of it. You start building upon this, you can get larger and larger three-dimensional structures. You look here, this is what the actual hairpin looks like. We draw it like this, when it's actually this twisted double helix looking thing that is actually one long strand as opposed to the anti-parallel that we talk about with DNA. You look here above my head. This is, it's better if you look in the textbook. This is tRNA. By going through and do this extensive one long strand of RNA that goes through and bends back on itself multiple places, multiple ways, forming these hairpin loops. And you look right here. This complementary base pair, it's not even from two sections of this RNA that are close to each other. This is further down the sequence that's coming back and binding here close to the terminal end. Goes back and forth, back and forth. This is what it looks like two-dimensionally, but when you put it into three dimensions, remember these hairpin loops are actually helices, you get this globular look. Now, I know in textbooks, they like to draw it where it looks like this. And I'm drawing this from memory, so it may not be totally right. It looks something like that. Some weird, somewhat cross-looking shape. I think I got that backwards. But anyways, it doesn't matter. The reality is, all these hairpin loops, which are drawn here looking like hairpins, are actually helices. And these bulges and bumps you see right there give bends so that it forms this highly complex three-dimensional structure that is actually quite stable. This will hang around for a while. This isn't there for just a few minutes and then isn't inherently starting to fall apart and all that type of stuff like we were talking about with messenger RNA. So you can get these highly complex structures based on the fact that this is one long strand, and one long strand that you're going to see here, you get one of those little triplets forming. So it's not, another strand inserting in. It's one strand that's forming this triplex, that Haagenstein or whatever the name is, association. A, both of them going to U, but not doing the same hydrogen bonds that you would normally associate with. You see it here again also. They're interacting. You get what? Ribose, 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 ribose. So you're getting 4 here. 4 It's the same RNA strand, but 4 different places are all interacting. Guanine, methylguanine. And then, some of the times, A to G, which is quite against Chargaff's rule, the A to the D, or A to the U if it's RNA, but this is a special guanine, N to dimethylguanine. The dimethylguanine means it's been methylated, 2 methyl groups, which alters its hydrogen bonding pattern. So as I said, it's not this simple RNA strand that we draw it. It's not that simple cloverleaf / cross-looking shape that we draw it as. It's this highly complex mishmash of triple bonding between nitrogenous bases, the hairpins that are actually helices of a single bent back in on itself RNA strands.